Hello students, this is Dr. Ranjini, Assistant Professor in Biotechnology. Welcome to e-learning sessions in cell biology. Today in this session, we learn about structure and functions of lysosomes. Dewey in 1955 named these organelles as lysosomes. Lysosomes is an organelle which provides an excellent example for for the ability of the intracellular membranes to form closed components. So, these are the membranous components. These are the closed membranous components which are filled with lytic enzymes, in which the composition of the lumen or the content of these closed compartments differs from that of our surrounding cytosol. So Dewey in 1955 identified the lysosomes and named them as the lysosomes. And these are nothing but the membranous, the closed compartments filled with the many lytic enzymes. But when the composition was checked between the cytosol and the lysosomes, there was lot of difference. The cytosol content was entirely different from that of the lysosomes. So lysosomes were filled with the lytic enzymes. And he found exclusively these lysosomes in animal cells. So lysosomes are present in the animal cells in the large numbers, and these lysosomes are responsible for degrading certain components that have become the obsolete for the cell or the organisms. So when the foreign substance enters inside the cell that is not required, and the extra waste products which are produced by the cell, or the dead cell, or the dead the remnants of certain cell organelles, that has to be thrown out of the cell. Such components which are not required by the cell has to be thrown out of the cell, and that function will be done by the lysosomes. Lysosomes are often budded from the membranes of the Golgi apparatus. So these are produced from the Golgi apparatus, but in some cases they develop gradually from the late endosomes. They are produced from the endosomes, which are vesicles that carry materials into the cell by the process of endocytosis. Endosomes are nothing but the invagination of the cell. So if you find the cell surface in the presence of certain extracellular matrix or in the presence of certain pathogen, it will show the invagination of the plasma membrane, and this plasma membrane invagination deepens and it will form the vesicle, and these vesicles are called as endosomes and usually filled with the lytic enzymes, and this process is called as the formation of the endosomes by the invagination of the plasma membrane is called as the endocytosis. Most of the time these lysosomes are formed by the Golgi apparatus, but sometimes we will find the lysosomes by the formation of the endosomes. The biogenesis of lysosomes requires the synthesis of specialized lysosomal hydrolases and membrane proteins. The lysosomes are nothing but the closed vesicles, and they require the formation, or the biogenesis of the lysosome requires the synthesis of enzymes like hydrolases and membrane proteins. So these membrane protein play an important role in exhibiting impermeability towards these hydrolases. So it's a bag of enzyme, hence it is called as lysosome is otherwise called as suicidal bag of the cell, and these hydrolases should not be leaked from the membrane. It should not be leaked from the membrane. So that's why during the synthesis of the lysosome, it requires the synthesis of the enzymes as well as the membrane proteins. Both classes of proteins are synthesized in the endoplasmic reticulum, and they are transported through the Golgi apparatus. They are synthesized in the endoplasmic reticulum and they are transported through the Golgi apparatus, and then they are transported from the trans-Golgi network. They will be bagged in the Golgi apparatus to an intermediate compartment by means of transport vesicles. So these proteins are synthesized by the endoplasmic reticulum. They are synthesized by endoplasmic reticulum, and they will be transported through the Golgi apparatus. And in the Golgi apparatus, there is bagging of these the components or the proteins takes place, and then they will be transported to their site of action. So here the endoplasmic reticulum is important, and also Golgi complex is important during the biogenesis of lysosomes. Coming to the occurrence, lysosomes occur in most animal cells, and they are present in very few plant cells also. They are absent in bacteria and mature mammalian erythrocyte. They are widely present in the animal cells, and in a few plant cells we find these lysosomes, and they are absent in bacteria and matured mammalian erythrocytes, or the RBCs lack these lysosomes. The few lysosomes occur in muscle cells or in acinar cells of the pancreas. So, they are present in the muscle cells and the pancreas. Leucocytes. Leucocytes, especially the granulocytes, are the rich source of lysosomes. They are rich source of lysosomes because these are involved in the defense reaction. These are involved in the defense reaction. Their lysosomes are so large sized that they can be observed even under light microscope. They are observed even under light microscopes. Leukocytes are they are the rich source of lysosomes, and the lysosomes are large sized because they exhibit high rate of phagocytosis, and hence they will be observed even under the light microscope. So this is regarding the occurrence. They are present in most of the animal cells and few plant cells. They are absent in bacteria and RBCs, and few lysosomes are present in the muscle cells and the pancreas. And the leukocytes, are the granulocytes which are involved in the phagocytosis, are rich source of lysosomes. They are the rich source of lysosomes, and the lysosomes are large sized and that can be observed even under light microscope. This is the structure of the lysosome. It is having the membrane. This membrane stability is very important in enclosing the many hydrolases, amylases, proteases, lipases, nucleases. All those will be enclosed within the membrane. And this membrane, it's a vesicular membrane that encloses the lytic enzyme. Along with that, it is also having the transport proteins to carry or to direct the movement of the lysosomes to the site of action. Lysosomal isolation and chemical composition. The isolation of the lysosome is very delicate. The lysosome is very delicate and fragile organelle. The lysosomal fractions have been isolated by sucrose-density centrifugation, that is Isopycnic centrifugation, after mild methods of homogenization. Take a tissue, grind it, homogenize it, and then you do the Isopycnic centrifugation or the density gradient centrifugation. You can separate the lysosomes fraction. The location of the lysosome in the cell can also be pinpointed by various histochemical and cytochemical methods, because this is a bag of hydrolases, and specific enzymes will be used for the used as histochemical marker to locate these lysosomes. And for example, lysosomes are uh, uh, positively reacting with the acid Schiff stain, and certain examples like certain enzymes are also good histochemical markers. For example, acid phosphatase is the marker enzyme to locate the lysosomal fraction. And for example, acid phosphatase is used as a marker for lysosomal fraction by the use of Gomori staining technique. So that's a simple technique which will be used to identify the lysosomal fraction. Specific strains are also used for other lysosomal enzymes such as glucuronidase, aryl sulfatases, etc. will be used. A lysosome may contain up to 40 types of hydrolytic enzymes. They include proteases, nucleases, glucosidases, lipases, phospholipases, phospholipases, and many lytic enzymes are packed in the lysosomes. The membrane of the lysosome normally keeps the enzymes in the latent phase and out of the cytoplasmic matrix or the cytosol. But the acid dependency of the lysosomal enzymes protects the contents of the cytosol against any damage, even if leakage of the lysosomal enzymes occur. The latency of the lysosomal enzyme is due to the presence of the membrane which is resistant to the enzyme that it encloses. So the proteins which are present in the membrane will be having the certain stability towards the membrane, and they prevent the leaking of these enzyme into the cytosol and further degradation of the cell content. Most probably the lysosomal hydrolases are membrane bound, which may prevent the active centers of the enzyme to gain access to sustainable groups in the membrane. They prevent the substrate and enzyme contact and prevent their action. The lysosomal membrane is slightly thicker than that of the mitochondria. It contains substantial amount of carbohydrate. The membrane of the lysosome is thicker than the mitochondria, contains the substantial amount of carbohydrate material, particularly salic acid. In fact, most lysosomal membrane proteins are usually highly glycosylated, which may help to protect them from lysosomal proteases in the lumen, because the membrane is also having the proteinaceous or the complex of the glycoproteins, and these proteases should not act on the lysosomal membrane. To prevent that, these proteases, the proteins are usually glycosylated and prevent the hydrolysis enzyme on the membrane of the lysosomal membrane. The lysosomal membrane has another unique property of fusing with other membranes of the cell. So it has the property of fusing with the other membrane, that is called as the coalescing activity of the membrane. So that is also exhibited by the lysosomal membrane. This property of fusion has been attributed to high proportion of membrane lipids present in the micellar configuration. Due to the presence of the membrane lipid, they exhibit coalescing activity. The surface active agents such as the liposoluble vitamin A, K, D, E and steroid sex hormones have a destabilizing influence, causing the release of lysosomal enzymes due to the rupture of the lysosomal membrane. So till now we were knowing the membranes are having the specialized protein. They maintain the stability of the membrane. They are called as stabilizer. But certain active agents such as the liposoluble the vitamin A, D, E, K and certain steroid sex hormones have destabilizing influence on the lysosomal membrane, and hence it releases the lysosomal contents into the surrounding. Drugs like cortisone, hydrocortisone and other tend to stabilize the lysosomal membrane and have an anti-inflammatory effect on the tissue. So cortisone and the hydrocortisone will prevent the leaking of this lysosomal content into the surrounding environment. The entire process of digestion is carried out within the lysosome. Mostly the enzymes act on the engulfed material in the acid media. The acidification of the lysosomal content depends on the ATP dependent reaction, and lysosomal membrane also contain transport protein that allow the final products of digestion, or after digesting the material inside the lysosome will be thrown out of the cell, or it is excreted out of the cell, or sometimes it is reutilized by the cell. So this is regarding the structure of the lysosome and the chemical composition of the lysosome. It's a bag of vesicle having the many hydrolases, and the membranes are made up of specialized proteins, and they stabilize the structure of the lysosome, preventing its leaking into the surrounding environment. They are called as stabilizer, but certain liposoluble vitamins and certain hormones, steroid sex hormones, can destabilize the structure and may lead to leakage of the content. But cortisone and hydrocortisone will stabilize the structure of the membrane and prevents the leakage. And that is regarding the structure of the lysosome. Next, coming to the functions, the lysosomes serve as the digestion compartments for the cellular material that have exceeded their lifetime. So, when the cell is aged, when the cell organelle is not functioning, in that condition lysosome will come into picture and it will act on the aged cell and will kill or it will digest it. Otherwise, no longer useful by the autophagy. So they will be engulfed and will be killed by the lysosome. That's why this lysosome is called as the suicidal bag of the cell. When a cell dies, the lysosome membrane ruptures and the enzymes are liberated. These enzymes digest the dead cells. So how these aged cells will be killed? By the lysosomes. When the cell dies, the lysosomal content will be, the membrane ruptures and the content will be liberated, and these enzymes, they start digesting the dead cells. In the process of metamorphosis of amphibians and the tunicates, many embryonic tissues, we find the action of the lysosome. For example, the gills and then fins and remole of the tail, etc. are digested by the lysosomes and they are utilized by the other cells. So these lysosomes play an important role in removing certain structure during the process of metamorphosis. Lysosomes break down cellular waste products and then fats, carbohydrates, proteins and other macromolecules into simple compounds, which are then transferred back to the cytoplasm as cell new building material. So macromolecules, not only the live cells or the dead cells will be digested, the lysosomes are also having the capacity to digest the macromolecules like proteins, carbohydrates, fats, etc. And they will be digested to their simpler compounds and will be transported to the cytoplasm for the further biosynthesis of the macromolecule. To accomplish the tasks associated with the digestion, the lysosomes utilize about 40 different types of hydrolytic enzyme. As I told earlier, it's a bag of enzyme filled with more of lytic enzyme. It is counted up to 40 different types of hydrolytic enzymes are present, all of which are manufactured in the endoplasmic reticulum and they are modified in the Golgi apparatus. So we need to prepare the hydrolases and the membrane protein for the lysosome biogenesis. So they will be manufactured in the endoplasmic reticulum and they will be modified or bagged in the Golgi apparatus and will be sent to the site of action. The digestion of the large extracellular particles. The lysosomes are involved in the digestion of large extracellular particles. The lysosomes digest the food contents of the phagosomes or the pinosomes. So, when the engulfing of the macromolecule takes place, they will be digested either by the phagocytosis or the pinocytosis. That function will be done by the lysosome. The lysosomes of the leucocytes enable the latter to devour the foreign proteins. So, these lysosomes are the rich in, or they are large sized in case of the leukocyte or the granulocyte. They exhibit the high rate of phagocytosis or the pinocytosis, and due to which it will engulf the foreign antigens or the foreign microbial protein like the bacteria derived proteins or the viral derived proteins will be digested or detoxified, and they play an important role in the defense reaction. Next is the lysosomes are also involved in the extracellular digestion. The lysosomes of certain cells such as the sperms discharge their enzymes outside the cell during the process of fertilization. During the fertilization, when the male gamete fuses with the female gamete, it will discharges the enzymes or it will secrete the certain enzymes that will digest the outer surface of the female or the uh, 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 female gamete and helps in fusion of the male pronuclei with the female pronuclei. That function will be done by the lysosome. The lysosomal enzyme digests the limiting membrane of the ovum and form the penetra path, or it will form a path to enter inside the ovum for the sperm. So this provide the space for the male pronuclei to fuse with the female pronucleus. The acid hydrolases are released from the osteoclast and break down bone for the reabsorption. So they are also involved in the breakdown of the bone and reabsorption. These cells also secrete lactic acid, which makes the local pH enough for the optimal enzyme activity. For the optimum enzyme activity, the particular pH is very important, that is maintained by the lactic acid, and the hydrolases which are released will be acting on the bone and they will be digesting that, and that will be reabsorbed in case of osteoclast. Likewise, preceding ossification, the bone formation or the fibroblast release the cathepsin D enzyme to break down the connective tissue. So during the breaking down of the connective tissue also, the extracellular digestion takes place by the lysosomal action or the hydrolytic enzyme. So that is regarding the extracellular digestion by the lysosome. So till now in this session we learned the structure of the lysosome and few functions. I hope you understood today's topic. Thank you.